Today, we will discuss about the overview of the proposed enhanced CAR T-cell immunotherapy against ovarian cancer cells that has been presented in the paper that we are discussing from the past few days. You can refer to the videos to understand individual experiments that are presented in the form of main figures. So, what is their final conclusions that they are proposing in order to increase their CAR T cells immunotherapy against the ovarian cancer cells? That first we will discuss what is the actual mechanism, how the immunosuppression occurs in these T cells. These are the T cells interacting with the cancer cells, and this T cells, when they are in the tumor microenvironment of the cancer cells, it will trigger ER stress, and this ER stress will trigger three signaling pathways. It is IRE1 alpha and XPP1 and also this ATF pathways. However, the pathways that cause immunosuppression is this IRE1 alpha. So what it will does is IRE1 alpha will cause this cleavage of this XPP1 mRNA in a way that it will form XPP1S mRNA, which is now functional, and then it will goes to the nucleus and it will inhibit this TAG-LN2 protein formations. The role of this TAG-LN2 protein formation is that it will interact with this FAVP protein in the cytosol and helps this FAVP protein to expressed on the cell surface, and in return these FAVP proteins, when they express on the cell surface, this will increase the fatty acid uptake, and so that the fatty acid will go into the mitochondria and it will perform T cells will be able to perform its function. However, because of the ER stress, this TAG-LN2 will not be able to secrete, will not be secreted in the cytosols, and thereby there is a problem occurs in the transfer of this FAVP into the cell surface, and the fatty acid uptake will go down because of that. And when the fatty acid uptake go down in the mitochondria, it will cause low fatty acid oxidations, because raw materials, that is fatty acids, are low uh, as this FAVP5 expressions goes down. This will cause mitochondrial dysfunctions, and it will finally cause the dysfunctional of the intratumoral T cells, as T cells will not be able to uh, do its regular functions against the cancer cells, that is killing the cancer cells. However, in order to overcome this problem of this low TAGLN2 expressions, what they did is they engineered a new CAR T cells, that is they named as overreactive CAR T cells, and this overreactive CAR T cells has this TAGLN2 expressions, so that these CAR T cells are able to, or we can say that overreactive CAR T cells DNA has this TAGLN2 complement TAGLN2 DNA in it, and so that will, what it will cause, it will this overreactive CAR T cells will cause overexpression of this TAGLN2, so that it can overcome the ER stress. Even in the ER stress conditions, the TAGLN2s are formed enough to move or helps this FAVP5 proteins to express on the cell surface, and um, thereby the situation that occurs when they use overreactive CAR T cells is that it interacts with the cancer cells and it will cause the release the enzymes, or release the cytokines that will cause leads to this killing of the cancer cells, such as granzyme, TNF alpha, INF gamma. And how this granzyme will trigger apoptosis in the cancer cells, we have discussed that thing in one of our previous videos, where we have discussed what are the different kind of apoptosis occurs, where there you can understand this granzyme, role of granzyme for extrinsic apoptosis. And these things occurs because of the overexpression of this TAG-LN2, and it can overcome the ER stress that cause suppression of this TAG-LN2, as in the overactive CAR T cells, the TAG-LN2 complement DNAs are included in this CAR T cells, so that the ER stress is not been able to completely suppress the TAGLN2 as in the case of normal CAR T cells. So I hope you understood the final summary of the paper that they have presented and what are the outcomes that they have find out from their experiments. If you need to understand this paper in detail, you can either go refer to our previous videos related to this paper, or you can refer to the paper directly from the reference link that has been provided in the description. So that's all for this video. Thank you for your time.